Okay, we'll get going. Good evening, everyone, and thank you for dialing in to the next episode of the 2024 State of Play series tonight. For those of you on Zoom, you'll probably see a notification about the meeting being recorded. Just click OK on that or agree to continue. And then firstly, we'll have an acknowledgement of country. MND Australia acknowledges the traditional custodians of country throughout Australia and their connections to land, sea and community. We pay our respects to their elders past, present and future and extend that respect to all Aboriginal and Torres Strait Islander peoples today. And I'd like to acknowledge the Wiradjuri people on whose land I am and pay my respects to elders past, present and emerging and extend a welcome to any Aboriginal and Torres Strait Islander people on this call. So tonight we're changing our format up again. As I'm guessing all of you are aware, we had our third annual Australian New Zealand MND Research Symposium about 6 weeks ago in Melbourne. And we thought it'd be a good opportunity to provide a bit of a recap from some of those who are able to attend, just to give their thoughts on what was good, maybe what could be improved, and what's hot and what's not at the moment in the MND research world. I'll run through the program quickly, and then we'll have some insights from a clinical care researcher, Rebecca Francis from Adelaide, someone with lived experience of MND, Taryn Hunt from Perth, and a lab-based researcher, Luke McAlary from Wollongong. Each of us will just give a quick 10-minute recap from the symposium, and then we'll have a joint Q&A at the end. Please fire any questions you have through the chat or the Q&A function if you're on Zoom, or if you're on Facebook, then just ask your questions or add your comments through the comment section. We'll collate all those questions and make sure they get asked at the end of the webinar during the Q&A. You don't need to wait to the end. If a question occurs to you, type it in the chat and we'll make sure it's we collect them all and ask them at the end. So I'll be starting off first. So I'm just going to load up my slides. So just bear with me a second. Okay, so as I was saying, we had the Australian New Zealand MND Research Symposium at the Florey Institute in Melbourne on the 27th and 28th of August. So just a quick overview. The program of the conference was developed by Tanuja Dharmadasa and Brad Turner, both from the Florey. And this was our biggest ever MND research symposium by a fair chunk. We had over 300 people, either in person or virtually. Had over 100 abstracts submitted, and we managed to squeeze 50 of those into presentations during the conference, with the rest being presented as posters. Of the attendees, over 30 of those were people with lived experience. And also we were fortunate enough to have a number of international rock stars, we'll call them, from the US, Canada, UK, Malaysia and Europe. This is courtesy of Fight MND, who are running a global roundtable which ran the 2 days after the research symposium. And they had flown in a number of these researchers in for their roundtable. A number of them were kind enough to extend their stay to participate in our conference. I think that probably contributed somewhat to the higher than normal numbers of attendees as well, because I think people always make an effort to see some of these global leaders present. We also had a number of the state association CEOs, and both CEOs and presidents from chairs of MND, Fight MND and MND Australia, and for representatives from MND and me. So we pretty much had the whole Australian MND community represented at this meeting, some aspect. So it was split over 2 days, the conference. The first day was pretty much more lab-based sessions, with day 2 being a little more leaning towards the clinic and care sessions. So the meeting started off with probably one of the biggest names in MND research, Professor Amar Al-Chalabi from King's College London, talking about what causes MND, providing a global update and new insights. And I won't say too much because I'm sure our other speakers will talk about this, but he did really integrates the environmental, genetic and other factors well into his talk. We then had 2 sessions with Australian speakers talking and New Zealand speakers talking about biomarkers and disease stratification, and then another session on genetics, genomics and cell biology. We then had a somewhat light-hearted debate around a very serious topic. So many of you might be aware that neurofilament light is becoming more and more prominent in many of our clinical trials. And the debate was, is this the most important biomarker in the MND ALS field? And we had Bob Blauser from Arizona and Andrea Malaspina from University College London as well. And against, we had our own Mary Louise Rogers and Nortina Sharizala from University of Malaysia. And look, the debate is basically NFL. Yes, it is the most important we've got at the moment, but we need more. It can't be a single standalone thing. We then had the rapid fire poster sessions. These were 1 slide, 3 minutes for each of the speakers, and chaired incredibly well by Lizanne Oy and Kathy Blizzard to keep all these presenters exactly to time. We had 20 presenters through there in a 2-hour session. The poster session then was in the evening and a bit of a mixer, and then we sent all the young, early and mid-career researchers off for a dinner to basically do a bit of networking. Day 2, we kind of split into 2 parts. We started off with Lucy Bruin, who's a Senior Director of Translational Medicine at Novartis in the UK, who gave a fantastic overview of where we are with the development of novel therapies for ALS, and our biomarkers, continuing that biomarker theme, are really critical for this. Continuing the drug development theme, we had a session on novel targets and drug development, as well as inflammation, immunity and neurodegeneration, where I think neuroinflammation is definitely a big target in a number of therapies being developed. We then had Shuan Ngo from the University of Queensland, who is an invited speaker, talk about the translational challenge. How do we move from the lab into the clinic, and are our models good enough for this? Following on from that, we then had another session on modeling disease. The second half of day 2 was really focused more on the clinic and care. And we started off with some fantastic lived experience insights from Phil Camden, who basically made everyone in the room, I think, laugh and cry at the same time. Followed by Professor Kevin Talbot from Oxford in the UK, who was talking about how we can target genes for new therapies of MND. We then had a session on treatment, care and clinical trials. And then we had a wide-ranging panel discussion. This was actually chaired by Mr. Iskandar Razak, who is a newsreader from ABC News in Melbourne, and he brought a really different perspective to this debate or panel discussion. And with panelists, we had various luminaries both from Australian and international, such as Matthew Keenan, Susan Mathers, David Berlowitz from Australia. Natalie Gould is a person with lived experience from New Zealand. And we also had Dame Pamela Shaw from the UK, as well as Jeff Rothstein from the US. Following that, we had another debate around targeted versus untargeted cocktail treatments with 2, again, luminaries of international research, Angela Genge from the Montreal Neurological Institute, and Dame Pam Shaw from the Sheffield Institute of Translational Neuroscience. And our final talk featured partnering with Plex, which a number of talks where we really focused on our increasing engagement with people with lived experience of MND in research in Australia. And finally, this conference wouldn't be possible without our sponsors. And we obviously have our main platinum sponsor with Biogen. We also have gold sponsorship from Teva, silver from Link Assistive, Pharmost. Lanyards are sponsored by Fight MND and LifeArc, MND and Me, Spinogenics, and Curalis were bronze sponsors. And again, it's great to see great sponsorship coming from pharma companies, showing that Australia is more and more involved in the clinical trial environment, and also there's more and more companies coming to MND to develop treatments, and I think that bodes well for the future. So on that note, I'd like to pass over now to Taryn Hunt, who will provide some of her takeaways from the conference. All yours, Taryn. I'll just stop sharing, and it should be all yours now.
Okay, thanks very much for inviting me to speak to you today. It's yeah, something I've not done before on behalf of a sort of a person without the scientific background. So I'll just share what I've put together, which is a couple of slides. It's nothing too onerous. And on the front, you can see that I wanted to give credit to End the Legacy, who paid for me to attend. And otherwise, it would have been a bit of a a trip from Perth for this event. So really grateful to them and for the work that they're doing in the genetic MND and frontotemporal dementia space. And that brings me to why I'm here at all, and that is because we found out this year that our family has the C9orf72 expansion mutation. So that has in my lifetime affected my grandmother and my mum. And I'm obviously at risk, and I have 3 kids who are also at risk. So it's suddenly become quite a significant thing for me. But before I do get too far into it, I just wanted to say that any views that I present are entirely of my own interpretation. So I may have, misunderstood or be misrepresenting anybody who said anything, to be fair, but I hope that what I tell you would be a fair representation of what we learnt. I also have a background in health promotion and also in medical librarianship. So I do know a bit about the scientific method, but boy, was there a lot of science there that just went straight over my head. So let's get into that. I did want to share a bit about, firstly, how it felt to be there, because I think that might be of interest. It's not something that you do every day, is attend a scientific conference that's outside of your field of expertise. And the first impression I had was, oh, wow, there's a lot of people here. And that was really exciting and uplifting. And I was really, thrilled. And I think it makes sense now that why so many people were there. And it was such a great one to attend, given that the round table was also on. But I had questions as well, lots of question marks, because some of the talks are really not for me, and that's okay, because I think there's quite a different audience in the room when most people are actually invested in the science. And of course, there was a little bit of emotional drain as well, because it's very personal when it's in your family, and I guess it just reminds you of that painful experience that you've had, and the fear that you have of what might be in your future. And I think that's something you have to really consider when you attend something like this, that, you might want to take time out, because it does, it can leave you feeling a little bit raw. But overall, I have to say it was a really positive experience. Oh, I went backwards, sorry. So what I observed were, sorry, I don't know if this one's going to work. Nope, you just get a blank slide, which is kind of annoying. Oh, here it is. That my observations, I think I grouped them into points that made sense to me. And as I said before, some of them might be a bit different to what somebody else would have taken away, but it seemed pretty clear that motor neuron, or ALS, is probably a multi-step disease. I think that came from Emma. So, it's a bit like cancer, where you need lots of things to come together to make somebody get the disease, and that exploring that is going to be really important. It's maybe some of the steps are covered by your DNA, but the rest of them are things that you could change, lifestyle factors or anything else that you could do, exercise or medication, etc. Yeah, we talked about, half of that went missing, biomarkers being really important. And while a lot of the detail was sort of over my head, it's pretty clear that we can't get much further without knowing when a disease has started and what progress, what triggers, what starts it. And I suppose this, with a DNA issue, you do want to know when things are starting to go wrong, because ideally, if you do know, you want to be able to prevent disease. But I don't think we're quite there yet. We did learn that there are some things that might be identified with motor neurone disease quite a bit prior to diagnosis. And it wasn't like that they were causative, but more like associated with. So things like antihistamines and steroids and things that affect inflammation, perhaps things that you might take for diabetes. But I mean, having said that, a lot of people take these for all different kinds of reasons. So it's hard to know how much that is of use. So yes, environmental triggers were one of the things that were talked about a fair bit as well. Lots more that we could learn there. And that, what happened? Treatments for genetic ALS or MND must be safe, given early, and reach both the brain and spinal cord. And I guess that stood out to me because, I mean, scientists know about the blood-brain barrier infinitely more than I do, but that a lot of things that we could do aren't going to reach the targets that we need. And some of this sounds pretty obvious, like that it should be safe, but it's not always. It's worth reminding ourselves of how important that is. And then this one is not my slide, and it belongs to someone else. I think the link is in the bottom right corner, or the information about where it came from. But I kind of liked it because it gave an overview for people without a lot of scientific knowledge about all of the elements that would make up a successful therapy. And I mean, to go back a little bit, we're not just talking at this conference about C9, MND, or SOD1, or any particular genetic factors, but we were talking about the full scale breadth of MND, so that it was difficult to know whether we would be aiming for something that is specific to one genetic mutation or something that might work across all of them. But so some of the things that we have to have in the bag, I suppose, are looking at combination therapies, so things that look at different pathways, preventative work, which is really important to people like me. We want to know, when does it start? How is it 15 years before symptoms? Is it 5 years before? What preclinical biomarkers are there? How would I know if I'm at risk, or if I'm going to be one of the lucky ones in my family who doesn't get it? That sort of thing. And then, yeah, some of the additional elements in there, I suppose, are just things to do with the neurons themselves. And there was a lot of talk about astrocytes and microglia, and I got the sense that some people spend a lot of time getting really focused in on those particular parts of cells. And I'm very grateful for that, but I can't tell you any more about it because I just don't know. But I guess that's really my summary. I'm not sure I could answer a lot of questions about the science that I took away from it, but I'm more than happy to answer questions at the end about any of my, sort of experiences as a familial at-risk person attending the conference. So on that note, I'll wrap up, and I'm going to pass over to Luke, and I'm going to stop sharing.
Thanks, Taryn. I'm just going to get my slides up. All right. All right, so I'm Luke, and I attended the conference, and I'd just like to thank the organisers tonight for this webinar. I think it's great that this is running, and also Taryn and the other people speaking for giving their insights that are also useful. So just let me, there we go. All right, so first of all, who am I? Right, so I'm a lab-based researcher, which means I work in a laboratory, with lots of different solutions and liquids and cells and proteins and all that. So I'm sort of your lab coat wearing mad scientist type of guy. I've been researching MND since 2012 during my undergrad years. And I obtained my PhD in 2017, and I've continued to research MND because I have a link to disease, and I really care about seeing this thing solved. So my focus is on proteins and biochemistry, so like the really nitty-gritty stuff that happens in this disease. And I currently work at the University of Wollongong, which has a very strong focus on doing MND research, due to Professor Justin Yerbury, who used to work there. And I'm part of a great team of dedicated people who are all driving towards trying to figure this thing out and trying to make new treatments for those who are suffering it. So I just want to give a little bit of an overview before I get into the recap to sort of tell people the different types of research, or the very basic types of research, I think, when you split them into two. So at the core of research is what we call basic or discovery research. And this is research that seeks to understand the fundamental aspects of some sort of observation or phenomena. And so this, for ALS, would be understanding the disease at its core. And the other type of research that's typically done is what's called applied research. So that's using knowledge to create solutions for problems, right? And this would be sort of like the development of treatments for MND. And so my favorite example for this is to why both of these go hand in hand, are important for each other, is quantum mechanics. That's a form of discovery research, right? And at its core, the strictest definition of quantum mechanics is that a small thing, very, 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 very small thing, can be both a particle and a wave, right? Doesn't make sense. That stuff's crazy. However, what that's done for us has allowed us to develop some of the most powerful computers in the world, right? So we have, every single person has one of these now, a phone in their pocket, because people decades ago did quantum mechanical research, right? So we didn't know what that research was going to give us, but it gave us some pretty powerful boons. And so that's why these two things go hand in hand now, because now computers are changing the research landscape and helping us do research even faster. And so the same thing can be said for MND. We need to do discovery research and applied research at the same time, because they both help each other. So breaking down what I learned at the conference into discovery research first, there were some key points. So first of all, is there support for discovery research? Because often you could probably argue that a lot of funding bodies in the world don't really do hardcore funding for discovery research, but I'm happy to report that, yes, there is, particularly via MNDA and FightMND. It was quite evident at the conference there are a lot of people trying to understand the fundamentals of MND, and this is really good to see, and I'm really happy to see that. The other thing I noticed about this was that, and Taryn sort of touched on this so did Gethin, is that the researchers are very engaged and driven. So there's not really a lot of I don't know, there's a lot of optimism and a lot of people who are passionate about this, which is, I think, really good. You don't want dispassionate people. And then the other thing was that a lot of research being done is exciting and clever too, which is great to see. And it's good to see from an Australian perspective, because I think our hosts were shocked. Australia is often overlooked when it comes to research, but they were shocked at how well and how good we were at what we did. Sorry, not our host, our guests from overseas. My bad. So here are my key takeaways. So one of the things that I thought was great to see was an increasing focus on PDP-43 function. So this is one of those main proteins that's involved in this disease. And there's been a sort of myopic sort of view of this protein will clump together and that's the only thing that occurs in disease, but there's now beginning to be a bigger holistic focus across every other thing that this protein does. And this is leading into both more better understanding of disease and also starting to bear some fruit, hopefully, for treatment. The other key thing that I thought was interesting to discuss with attendees was the fact that, or not the fact, but the concept that we might have exhausted our genetic data. Probably not completely, but it's getting harder and harder to find new genes because they're probably becoming rarer and rarer, and there's probably more than one gene implicated in certain patients. But it's a big thing, and it's not bad. It actually might be good. It could mean that we've sort of discovered the pathways that are involved, and now we just need to dig in and start figuring out how to get at them. The other thing too, and Taryn touched on this, is what environment and life factors contribute to MND was a big question. And it's been a question for a couple of conferences. And I think it will remain a question because it's a really, really hard thing to get at. It's really difficult to figure out how someone has lived their life, where they've lived their life, and how that may affect them. So I think in this instance, this is a time-based thing, unfortunately, where we're just going to have to wait to get enough data to pull out more and more things that may be associated and start to test them in a lab/clinical-based setting to see if these things are actually related to MND. And then a great thing to see, and you typically see that at this conference, is that there's always a further unraveling of multiple MND mechanisms. So it's, some of the lab groups, what they do, and every year you see them taking the next step, the next step, the next step, and it's really good to see that. And these things include understanding, how inflammation contributes to the disease, how oxidative stress is contributing, how protein aggregation is contributing, and people are making advancements all the time, which is excellent. So applied research at the conference, key takeaways. Improvements in delivering drugs seem to be on the horizon, which is great. And as Gethin and Taryn touched on, you can make a drug that will pretty much do what you want it to do, but you need to get it to where it needs to be. And that's always been difficult for pretty much any disease, but particularly neurological disorders, because your brain and spinal cord is protected by a very important physiological component of you, which is called the blood-brain barrier. And getting through that is necessary to get the treatments to where they need to be. And a lot more people are starting to look at this and work on this and show some pretty damn good success. So that's really good to see. An increased focus on personalized medicine. So this means treating the patient as an individual. And I think this is going to be absolutely necessary if we are to attenuate this disease. It makes running clinical trials very difficult, but it's becoming increasingly clear that every patient develops disease in a different way or from some sort of different start point. And understanding this is going to let us develop very effective treatments, hopefully. The other question, and Gethin touched on this, is are our current biomarkers good enough? And I think the explanation Gethin gave simply was correct. It is, yeah, we've got a biomarker, which is excellent, but it's still not good enough. And what is great to see is that people are working on developing new ways to detect disease, trying to figure out as early as possible, trying to make these diagnoses as quickly as possible so treatment and care can start as quickly as possible. And then the other good thing about the applied research was that there were many early stage therapies in the pipeline. So these aren't things that are going to come out next week, next year, but seeing that people are thinking into the future is, really good to see, and that they're thinking very creatively about how they may attenuate this disease. And they're taking into account all those previous above points too. So where do I think the gaps are and how might we have a look at this? So I think one of the major gaps that we as the basic researchers need to start trying to understand and be honest about is a failure of stem cell models to recapitulate disease. So we can make stem cells from patients who have the disease, but there's really a difficulty in actually seeing any type of readout that says that there's some toxicity or something wrong with the cells. we really need to dig deep into this and try and understand why. And this might tie back into that environmental stressor trigger thing. So if we can find a trigger, maybe we can use that as a better way to model this in the cells. Animal models are unreliable for predicting drugs. This is a common theme across biological science, and I think this is something that a lot of people in MND are taking on the chin and really trying to move past these poor animal models that are just not producing accurate information for what we think might be happening in disease and what we think might be happening when we treat the disease. So it's great to see people admitting this and starting to think better, trying to make better models and design things so that they're more accurate and more reliable. Like I was saying, specific biomarkers are needed for treatment and disease type. So we need to know when someone gets disease, and we need biomarkers that tell us how someone is receiving a treatment, right? And so there's some good stuff on the horizon for treatment-based biomarkers and disease-based biomarkers that I hope will provide some very fruitful outcomes, right? The biggest problem, as I see it, though, is that we are still modeling the disease as we observe it. And what this means is that scientists, we often come in like detectives after the fact. So we'll look at someone's who's passed away, and then we'll look at their tissue and say, oh, this is what must have, been the cause of their death. But sometimes that's not true. Sometimes that's a sort of side pathway that happened, and there's something else happened. And I think we as a community in the field really need to be open to new ideas surrounding how things are occurring prior to what we see in post-mortem tissue. And so what I've got bolded there, we need to model what causes MND, not just what we think is associated with MND. And so with that, I'd finish up and thank everyone for listening, and hope you learned something, and hope you've got some great questions later on for me to answer. And I think I'm passing off to Rebecca.
Yes, hello.
Right, let me stop my share, sorry.
Thank you so much. And I'll share mine. So hopefully you can see my screen. And if I just hit that, you should be able to see that now, hopefully. So I'd also like to thank Geffen and the organisers within MND Research Australia for having me at the conference, and also for inviting me to speak today. It's a great pleasure. Thank you also, Taryn, for sharing your thoughts and insights. And yes, a lot of the science went over my head too. It's a lot to take in. I'd also just like to mention that I'm presenting to you from Ghanaland in Adelaide today. So firstly, just a little bit about me. I'm a speech pathologist and an academic, and I live and work in Adelaide at Flinders University. And I'm 2 weeks away from submitting my PhD project. And I've been investigating the interaction between dysphagia or swallowing problems and cognitive and behavioural change. And I've just included a couple of published articles and the QR codes on the slides if you're interested in having a read of those. So my focus is really on improving care in MND, specifically around sort of what we do as a speech pathologist, but also care more broadly. So my PhD sits in that qualitative research space where I've done lots of interviews and lots of speaking with people with lived experience. So some of the research highlights for me, I think I'm going to echo a few of the things that have already been mentioned. So Amar's talk was amazing, and it helped me learn so much about the disease in that, he discussed this multi-step model. So these six steps, and that genetics is kind of maybe the first couple of steps, but we need that interaction between the environment to trigger the onset. And so some people may never have that environmental trigger. And I think also what they talked about, and what I've since read after the conference, is the pedigree and the importance of understanding the greater family, the genetics of the greater family. And so what might look like sporadic can be related to genes somewhere in that larger pedigree. And so I guess what we took out of it was that MND is really a complex disease with complex mechanisms, and there's not one cause, similarly to cancer, and therefore it's not really just that one disease. Some of the fascinating science that I took away, obviously there was a lot about TDP-43 and a lot of it was quite over my head, but one of the things that I found really quite fascinating was the idea that we can potentially recreate a corticospinal tract. So I just got a little photo that I took of the presentation on the day, and I've just included a picture of essentially what is the corticospinal tract. And I think it's really important that, because we know that motor neuron disease targets the whole system, it's important for us to be able to look at the whole system in research. Obviously, I'm speaking as a clinician, not as a lab researcher, but to me, I found that really interesting. So if we can recreate this lab-based corticospinal tract, what will that tell us about disease mechanism? The other one that I found really interesting was a care talk that came very late in the day on the second day, and it was about exercise in motor neuron disease. And I found this really interesting because there is some literature that looks at the sort of contraindication, or the idea that exercise is probably not a good idea, because we know that weight loss is something that's not great in MND. But it was looking at, exercise as medicine, but really making sure that we get the dosage right. So I did find that presentation really interesting, and I really look forward to reading that article when it's published. One of the things that I found that speaks really to my heart is partnering with people with lived experience. We know that there's barriers around research, and it's purely the connection of people with researchers. And so being able to have a lived experience network that's recently launched, where people can sort of put their names down to participate in, really bridges the gap. And it allows people that are wanting to conduct research and people that are wanting to be involved in research, which, after my experiences of attending conferences and speaking to people with MND, overwhelmingly they want to be involved. And so we really need to look at, really strengthening these connections between researchers and lived experience, so that we can create more data, because data sets and big data sets are really important in MND. So we need to be able to, share information as researchers and share data sets. And a lot of that is happening with the MND collective that I'm involved in. And so there's some really great work at sort of joining the dots and bridging those gaps. Phil gave an absolutely beautiful talk at the end of the presentation that really spoke to my area of research, and it was talking to the panel about, whilst there is no cure, how can we improve life? And this is exactly what I am aiming to do and wanting to better understand. And I think to do that, we need to understand the preferences of people that are living with MND, both their carers and the people that are experiencing the disease. And it's so important for us to listen to and learn from people with experience. And I certainly am hoping to conduct some more co-designed research projects that look at investigating what's really important and linking those, linking people with lived experience and understanding their preferences. So that's my bit of information. So I'll hand it back over to Gethin, and more than happy to answer any questions.
Thank you, Rebecca. And thank you to all our presenters. If you could all bring yourself back online. Those were fantastic insights to the conference. And honestly, Taryn and Rebecca, I spent 20 years in a lab, and most of this stuff still goes over my head. So I think the level of research we have in Australia is incredibly high. And just mirroring what Luke said, I actually spoke to a number of those sort of superstars who came in from overseas, and every single one of them said they were really, really impressed by the level of research. So it's a fantastic opportunity for us to showcase our research right across the board to all these people. So hopefully it'll be hopefully they won't go and steal them all and take them overseas. But I think it helped us a lot in that respect as well. So a number of comments come out of that. things I noticed, again, I think we agree that the whole cocktail versus targeted treatment, I think, as Luke said, I think the idea that it will be a question of how we can identify what exactly has gone wrong for each person. There's different pathways, there'll be different causes. And when we can start identifying and putting those apart and say, okay, we've got, say, 10 drugs that could treat different aspects of MND. And I think when we can say, okay, you need to take 3 of these that will suit you best, I think that will be absolutely brilliant. And that will take us forward. Again, we have to get the drugs to people in the right space. So, you know, it isn't easy to get them in. Unfortunately, with the genetic therapies at the moment, it's a monthly injection into your spine. I mean, they work, so I guess that's a price worth paying. But we're hoping in the future that they will get these drugs will either be a single hit, you know, in the future for a genetic therapy. You know, you'll put itself into your genome and you're one and done, or even just once a year or something. So I think that's where that's going, hopefully. And the biomarkers, yeah, it's really important to know to diagnose the disease, but also great to have biomarkers to tell us what pathways aren't working, and you know, to give us maybe some prognosis. Is your disease going to go really fast? Is it going to go a bit slower? And is this drug helping? So we've got a whole bunch of questions that just come in now. So I don't know if Taryn or Rebecca or anyone here had a little bit more insights into the exercise challenges. You know, what's good, what's the correct dose? I think the researcher is Alexandra Ferry, I think is the researcher. So we can put some links up to there after the talk to their work. I don't know if Rebecca, you had a few more insights from that.
I did a very, very quick deep dive to try and find it before jumping online today. So I'm happy to have another look and send you what I find if you would like me to do that, and then you can share it. But it's an interesting one because, as I said, there's sort of this train of thought that it's a contraindicator, but it's also I found her talk really interesting. So I'll be happy to do a bit of a deep dive and try and find that one.
And we've got a question here, which has the conference generated any specific adjustments to MND Australia or Fight MND's funding priorities? Obviously, I can't speak to Fight MND, but it's a question I might ask you three is, was there anything obviously lacking, you think, in the research? Is there something we're missing that we perhaps should be doing in Australia or be doing better? Is there a gap there where we could identify a priority? Sorry to put you on the spot, but there you go, you volunteered. So
from my point of view as somebody at risk, I keep coming back to technology as the potential, CRISPR technology, just cut past all the trials of other things and go straight for the big guns. But I got the impression that from the conference that it's both costly and we're just not there yet in terms of human trials. And my recent sort of look into it was that only a handful of people have been successfully treated with CRISPR for anything. So I guess that's probably something I'd like to see a bit more activity on.
Yeah, I think I'd agree. I think, yeah, you're right in that CRISPR is very new technology. And again, we also have to know exactly how to do it. I mean, unfortunately, there's been 2 C9 trials that have failed, both which people were not confident but very optimistic about. So we sometimes have to understand the genetics more than we thought, I think. I mean, the SOD1 trials seem to be working really well. Loose looks positive, but the C9 is more complex, unfortunately. But as you saw from the conference, there's a heap of very smart people trying to work this one out. Anyone else have any comments on that?
Yeah, I've got a comment on that. So I think we do a lot of, and I'm probably shooting myself in the foot here, but I'm a biologist, and a lot of our research is focused on biology. I think one of the gaps, and I think an absolutely insane growing area of science, is brain-machine interfaces. And I think those are sort of things that can help patients now, right? Being able to take control again of some sort of prosthesis with their brain, because most of the time someone suffers with MND, they can't move, but they can still think clearly and cognitively. And those sorts of things, I think, are probably more immediate than any biological based therapy that we're gonna generate. So I think a little bit more engineering would be interesting to see at these conferences, a little bit more mechatronics and that type of thing.
Thank you, Luke. Question here to answer Keith's question in more detail, I don't know if we found new funding priorities, but I think the takeaway is that it's definitely identified perhaps areas we should focus on and areas of strength in Australia as well. I mean, I think biomarkers and data is undoubtedly an area where we need to keep working, and that can then feed into everything, I think. A question from Hannah is there a list of known biomarkers or potential biomarkers that have been investigated? They're certainly out there in review papers. We can certainly dig some out and make them available to people, what the information is out there. I don't know if Luke, have you any other insights in that?
I'm not a biomarker researcher. Yeah. There definitely would be people making lists, and I think it is a good idea to maybe make an easy to access compendium of them. But often you don't know what people are working on until they report it. But people are definitely working on them.
Yeah, and that goes to I think the what actually came out of the global roundtable that followed the our symposium was a desire of people around the world to work together to make sure we're collecting data that is compatible, and that can be make sure we always collect data in the same way and the same data from no matter where you are in the world. And that goes also to Peter's question about how we can beef up our data collection. We have data collection in Australia. MND Australia is currently trying to work with the government to persuade them to fund a national data collection that goes beyond just the simple patient data, to collect data from the state associations. So we have data about care models, about what drugs that people are taking, what treatments they're using, to really build up a really complex picture of where everyone is in Australia with MND, and possibly also identify environmental factors and how they're dealing with it and where the care needs are. So that is something we're really looking to. And what we really need to do is to make sure that data is compatible with other organisations around the world, so we can share it and pull it together. I'm just scrolling through. Everyone needs a Luke. There you go, Luke. Comment of the night. Okay. So we understand that about 90% of the disease is sporadic, but there is an increasing awareness that there are more and more people do have genetic causes to it. I think you all identified that from the disease, especially from Amar's talk up front and Kevin's later on. If anyone else wants to comment a bit more about that, about the how genetics is becoming more and more critical in MND.
I think the thing that I took out of that was the pedigree study. So when you're looking at, mother, brother, sister in that small sort of family unit, but it's getting to the point of understanding second, third, fourth cousins, and often we don't have contact with, those relatives with it, and so therefore we lose the knowledge that this might have happened in another family member sort of quite distant from us, but the gene is still obviously as part of that bigger pedigree. So while it appears sporadic, there are potentially some familial links that are not known.
I thought he was also saying that it's important to study genetic carriers because they already have several steps covered. So in a sense, you're part way there and you've only got to identify the remaining few. And when I say that, I realise that sounds a lot simpler than it would be in actuality, but that was my impression and I think, you know, speaking as someone from that point of view, anything we can do to help, it just gives you that sense that you're going to leave, you know, you're going to add something to the process. So yeah, please do reach out to us because we all want to help.
Yeah, I mean, I agree, Taryn. That was quite striking. The idea is that, yeah, if you have these 6 steps and perhaps certain mutations might count for 4 of them, then you only do have to identify 2 other potential environmental factors rather than perhaps 6 in some people who aren't carrying mutations. So I won't say it simplifies it, but it certainly could make it a little more straightforward. The other problem, obviously, with the whole family is some people don't realise that, you know, Uncle Sid or whatever did have MND and perhaps, you know, died of something else before anyone ever knew he had MND. So I think, yeah, it's very challenging to get really accurate genetic histories in some families. Where, you know, it's not always clear what's happening. One question I have wanted to throw out to you. Did you have a single highlight from the conference at all? Was there a single talk that really you went, wow, that was brilliant, I'd watch that again, or really struck you as really insightful? I can go first. For me, it was Amar. I'm a bit of a fanboy of Amar, I'm afraid. So I've heard him speak before, but every time you learn more. He's really presents those big concepts well.
Yeah, it makes it very complex, very simple, I think, and very digestible for everybody that was there. And I think listening to Phil for me was a highlight too.
I forgot to mention and I think Luke mentioned this, the breadth of creative ideas that were presented in the posters. I was blown away by the different interventions that people were trying, even cold therapy, to, you know, gene manipulation and you know, diet and exercise, and you know, it's something we know in the library and research world as well, that you have to try things and report what doesn't work so that we all learn from that experience. And I think I was really encouraged that people talked about what hadn't worked. As depressing as it might be, at least we know now. I thought there was a lot of honesty and just yeah, real passion just to get to the bottom of it.
Yeah, I was definitely a fan of Amar. But I think my big highlight was seeing the rapid fire talks from all the young people who were coming up in the field and seeing that they were extremely effective at communicating their ideas. And also these ideas were really, really good, which means that the future of the, you know, research community as a whole, including the lab-based and the clinical-based, is pretty strong, which I'm really happy to see. There's more and more enthusiasm every year, which is what I like to see.
And just going back to some of the talk, Peter Roddick has just suggested that there's an organisation in the US called the ALS TDI who run a data program called the ARC. I'm just trying to remember. I think it's the ALS. I can't research. I can't remember what the C stands for, but that is a big data collection that people can do remotely from Australia as well. You can contribute to it, even though you're not able to visit a site in the US, and they have set up to remote collect people. Even though we still want all our Australian patients to obviously contribute to MindOz and our databases here, people can also contribute internationally as well to boost that data around the world. Oh, Phil's online as well and says thank you as well. Yeah, we recognise that, Phil. That was a fantastic talk you gave. Right, we're coming close to the end of the time now, so I just wanted to wrap up and say a couple of extra things about how appreciative I am of everyone coming along and giving your thoughts about this. I know it's kind of, you've been put on the spot a bit with asking you your takeouts from a conference, much of it not in your own areas, but I really appreciate it, especially Rebecca, 2 weeks away from finishing your PhD. That's quite impressive. I think most of us at that stage were just running around like utter headless chickens to get everything in. So congrats to be able to put something together. And Taryn as well, really appreciate you coming on as a, you do have your science librarian training to come into an audience like this and put yourself out there. We really do appreciate it, and I'm sure we'll be engaging with you lots more over the next few years with the MND Research Collective, and hopefully many of the people online as well will engage with that and the lived experience network, because I think it's really critical that we all work together. I really like what Luke was saying, basically that should guide our research. What research do people want? What do you think is going to work if you are someone with lived experience of MND? And I think those opinions, they may not be able to tell you exactly how to run your study, but they can certainly tell you whether your study makes sense or not. And I think it's really important we get all those voices in. So thank you to all three of you again. And like I said, I'm sure we'll be reaching out many, many more times. There's a couple of comments up in the chat about some questions, and we'll try and make sure they're available on our website as well, because I think the chat disappears as soon as the webinar finishes, but we'll make sure then that information is available as well. So thank you again to everyone. Much appreciate your time, and we'll be back in a month or so with the next state of play. Anyone who registered tonight will get a link to a recording. So feel free, if you want to watch again or pass it round to your network, let them know it's available. And also our previous recordings are also available for all the other state of plays, as well as the recordings of most of the main speakers at the symposium are up on our YouTube channel. So if you go to the MND Australia YouTube channel, feel free to watch some of those that we may have highlighted tonight that piqued your interest. So good night to everyone. I'll let you all go off and have your dinner or enjoy your evenings, and thank you all again.